it has been aired also. Yeah. So I'll share my screen. Simultaneously, I'll introduce myself once we get to the presentation. Yeah.
गांव का एक वाक्य मैंने कहा ना छोटा ये गांव का कहता साउंड तो रहेगा
Okay. So I hope my screen is visible to everybody. If at any point during the lecture, multiple of you think that the screen is getting stuck or it is like you can't view the presentation, it shouldn't happen because I'm connected to the institute Wi-Fi. But in case that happens and in case that misses my notice, please let me know. As for an introduction, so last Monday I was unable to take a class, which is why I had to postpone it to Friday, which is when I think all of you are new names for me. So none of you at least had joined on Friday. Uh, so this is the second lecture that I am taking. I'll upload both of them on the on YouTube, and I think the link will also be shared to you by NPTEL. So as for myself, I am Arveen. I am currently a PhD scholar at IIT Kanpur, and I am also pursuing my PhD in the field of neuroscience. I am working on a neurodegenerative disorder, and I am trying to understand its relationship with uh, vasculature and how there are deformities and defects that form in the blood-brain barrier, which could in turn impact the brain. So that is my line of study. So which is what has gotten me interested in neuroscience in the first place. So before my PhD, at least, I have been a graduate and postgraduate in zoology. I have not had neuroscience per se as a subject. I have studied a lot of physiology, anatomy, uh, in humans, animal sciences, and human sciences, but neuroscience per se was a new subject for me when I joined PhD. So, but for the past 5 years, I have really been understanding various aspects of neuroscience, and this course provides another insight into that aspect. So I am also learning along the way, but yes, there are a few things that I also have to add to the course, which is why we'll be conducting this session, right? So what I'll do each week, and I have scheduled my classes for Monday of every week, so 7 to 9 on Mondays. I'll be taking this session wherein what I'll do is I'll go through the previous year questions pertaining to the week whose submission is actually due. So, for instance, I could see that your week two submission is due in 2 days, right? So, before that submission is done, I can't really get into solving those particular questions with you. But what we can actually do is we can solve the previous year questions. Pertaining to this week, at least, it is mostly about history and who did what. So that anyway, if you have gone through the lectures even once, you will have the answers to that. You might have to go back again to that particular lecture in which that particular thing was being talked about to refresh your memory, but the answers will be pretty straightforward, right? So, but as and when we go uh, deeper into neuroscience and concepts of neuroscience, the questions might not be that straightforward, which is why we do this exercise wherein we solve the previous year questions, which will probably help you get better understanding or gain insights into this year's assignment as well, right? So that is going to be the first part of the discussion. As per the second part of the discussion, what I try to do is either add on to the course that actually has been taught to you, or if there are some things that were touched upon, but I feel being new to neuroscience myself, like if I picture this as me being a student 5 years ago, I would want a little more understanding or knowledge about a few topics. So I try to expand those topics and talk a little bit about them to clear our basic concepts. And uh, else, what we'll be doing, at least today, is going through a different perspective of history of neuroscience, which is mainly based on Candle, which is this uh, Candle's book that is this Principles of Neuroscience, right? So Candle was again a very renowned scientist in the field of neuroscience. We'll learn about him later. So we'll discuss about that part of history when we come to it. For now, what we'll do is we'll begin with the previous year questions pertaining to week two. And what we'll do is we'll first read the question, we'll read the options, and then eventually we'll come on to what the answer is. And if that answer requires any additional knowledge or insight, we'll be discussing that, right? Also, you guys are free to switch on your mics and to ask questions if there is a doubt, right? Even if I can't solve it right away, I will try to read upon it. I will try to ask my professors and to get back to you, right? So that is what I can help you with over here. Okay. So, beginning with the first part. Yeah. So, who first observed neuronal development using a microscope? Right. So, this was the first lecture of week two. Right. So, was it Ross Harrison, Wilhelm Hayes, Ramonica Hall, or Hans Spemann? Right. Sorry. So, the answer to this is Wilhelm Hayes, right? So, no conceptual knowledge required in this. This was a memory-based question, right? What Wilhelm Hayes had actually done was that he had taken multiple kinds of embryos at different stages of their development, and then in these different embryos at different time points of development, what he had done is microscopically he had viewed the neural tube development and the axonal development, right? So that is what gave him some insights about how this neural tube actually develops. Then again, the other one is, what does NGF stand for? Whether it is nerve growth factor or neural growth function or neuron growth factor or natural growth factor, right? So in this context, we know that it stands for nerve growth factor, right? Which is now uh, well classified, described, uh, uh, chemical which is present in our nervous system, the nerve growth factor, and it basically, yeah, okay. So, what did scientists do in their early experiments to understand axonal transport? Right? So, how in their early experiments did they understand how axonal transport was actually happening? Right? So, did they paint the axons? Did they tie or ligate a neuron? Or they took pictures of the axons? Or they counted the nerve cells? Right? So, out of these, actually, what did happen was that they tied or ligated a neuron. So how did that happen was, so first off, to begin with, this long part of the neuron is the axon, right? This is a part that is coming out of the cell body or the soma of this neuron, right? So this axon, what they did was they wanted to understand how transport used to happen in this axon, whether it used to happen both in both that directions in one direction, whether there was actual transport of material that was happening from say this point to this point, right? So all this the scientists wanted to understand. So what Paul Weiss actually did was he took this single neuron, and what he did was he tied or ligated the neuron along some region of the axon. Right, what happened was that once he tied this region, he saw that just before this region there was bulging at the site of this ligation, right? Which meant that earlier there might have been something that might have been flowing from this region of the axon to this posterior region of the axon. But now, after this axon was actually tied with a rope or a strand of a hair, what happened was this material got collected towards the region before the knot, right? So that is why there was bulging in at that region, and the terminal region or the posterior region started degenerating because it did not get that material that was required for it to survive, right? So this is how axonal transport was actually initially studied. Also, like this happened much later in case of neurons. But if any of you have, and we all might have at least at the level of 10th or 12th, we have studied plant biology as well, right? So if you would remember, these kinds of experiments had already been done long ago in case of plants, right? Wherein the transport through phloem, xylem, etc. had been studied in a similar way that either some part was shaved off and it was seen that the top region and the lower region there was bulging that was observed in case of the plant because now water or nutrients were not flowing through this one region to the other region, right? So these kinds of experiments had been performed in a separate system altogether, right? But like over here, the scientists were accredited with actually applying that concept into a human neural cell as well, right? So that was a major part. Okay. So question number 4 is, what did Claude Bernard discover about the arrow poison curare? Right? So was it that it worked instantaneously when applied to muscle cells? Or it was used as a medicine, or it was completely harmless to animals, or it caused paralysis when applied to muscle nerve cells, right? So, while he was studying this particular poison, curare, what Claude Bernard actually observed was that it caused paralysis, but this paralysis was restricted to when it was applied to the muscle cells, muscle nerve cells, right? So, what happened was he tried it both ways, right? So, for example, over here you can see this is a representation of a muscle fiber bundle, right? So these are the muscle cells, and this is a neuron that is innervating this muscle, right? So what Claude Bernard first tried was that he gave this poison curare only to the muscle cells, right? But it did not have any effect like paralysis on these muscle cells. However, when this poison was actually provided to the nerve cells which were innervating the muscle, it resulted in paralysis of the muscle, right? So this was what was actually observed. Then the question is, who first talked about the lock and key idea, right? Was it Otto Louis or Charles Sherrington or Emil Fisher or Walter Dixon? Right. So again, this is a topic that is not specific to neuroscience per se. Even in molecular biology, it is very well renowned. That Emil Fisher was the one who first talked about the idea of lock and key. Right. Now, what he proposed was in 1894 was that the substrate and the enzyme they fit each other like a piece of puzzle, as if this enzyme was the key to a lock, right? So generally a lock and key are a very specific relationship, right? So one key will probably open just the lock that it is designed for, right? It will not like those TSA universal or master keys that they have at the airport that can open any TSA lock, right? It will not be like that. So traditionally, one key is that is designed for a particular lock will only open that lock, right? So that was what was proposed in terms of substrate and an enzyme also, that they will fit each other like a lock and a key or like pieces of the puzzle, right, coming together. Then the next question was, what was the key methodological advancement in neuroscience during 1950s? Right. So this is an interesting one actually. Was it studying brain activity at the organ level or developing the glass micropipette as an intracellular electrode or creating textbooks about brain biochemistry or identifying only two neurotransmitters? Right. So just how the statements are written. Also, you can, just a moment. Yeah. You can negate out one or two, right? So, for instance, you know that brain biochemistry was not that big a thing in the 1950s, right? Still, the most of the research that had occurred was at organ level, right? So this was happening. The first case organ level study of the brain activity had been happening for a while now. You're starting from the 19th century itself, so 1800s and all. Studies were happening at organ level. There was nothing new about it at in 1950 also, right? Biochemistry textbooks, um, the relevant ones came a decade or two after 1950s actually, right? Identifying only two neurotransmitters, right? Itself, it is like a demeaning statement that only two neurotransmitters were found. It is said only like that, right? So you can negate this also for now. Developing the glass micropipette as an intracellular electrode was what was actually interesting, and it was a new finding which was very relevant at this era, right? At this time. So, yeah, who actually discovered this glass micropipette and this concept? It has been, accredited to being co-invented by Gilbert Ning Ling and Ralph Waldo Gerard. Right? Gerard had more to do with it than Ling. Why we'll understand as and when we read this text, right? One second. Yeah. So, what this particular statement is uh, actually fine, sharp-tipped examples of capillary glass microelectrodes had been developed and used successfully back from the 1920s, right? But mainly it was used in plant cells. Again, another example of how that ligation had already been, practiced in plant cells, and now it was being extrapolated in neurons, right? Similar was the case with this glass micropipettes also. So Girard was, however, involved in the successful transfer of their use to single skeletal muscle cells, although his participation in that too occurred long before his much cited 1949 paper with Link. Right, so 1949 was when he got recognition, and that was a paper that he had published with Link. Right, but before that also he had actually began to use this in single skeletal muscle cells, right? Also, by the late 1930s, Gerard had developed a keen interest in the possibility of producing microelectrodes to use in individual neurons to monitor their electrical activity. Right? This is actually possible right now. It is a very famous, very accurate method, but very highly invasive method, which is known as patch clamp, right? Through which the voltage of the resting membrane, etc., had also eventually been tested, right? It was using this kind of a technique only. So this is an example of that voltage testing, right? How we came to actually know what is the resting membrane potential, how there is a potential difference between the outside and the inner side of the axon, right? All these things we came to know uh, uh, via this kind of an electrode that was used. This is an experimental snapshot of the Hodgkin-Huxley experiment on the giant squid axon. And this is known as a giant squid axon actually because it has a diameter of about 100 or oh sorry 1.5 mm around it has the diameter. 1.5 mm is something that we can see with our eye, right? Our neurons, they have diameters ranging in microns, so you won't be able to see it, much rather work with it in this manner, right? That is why the squid was a good model organism for study of these membrane potentials in neuroscience, right? So this glass micropipettes were what were actually used to do this study. Yeah. So this is a representation from the study that Hodgkin and Huxley had done. Let's read the physical legend. This historic tracing is the first published intracellular recording of an action potential, right? So it was recorded in 1939 by Hodgkin and Huxley from a giant squid axon using glass capillary electrodes, right? Similar to this, what we have been talking about. Filled with seawater, the timing pulses are separated by 2 milliseconds. The vertical scale indicates the potential of the internal electrode in millivolts. So this will be plus 40 millivolts. And the seawater outside being taken as zero potential, right? So, these kinds of studies, also the first action potential, for instance, was determined using this kind of a technique. Yeah. Okay, moving on to the other question. What did Ramoni Kahal call the tip of the axon? Whether he called it the nerve tip, or the cell connector, or the axonal growth cone, or the neural point, right? So, the term that he used was axonal growth cone, right? Why I have kept this image on the right is because these are actually hand-drawn images of the neurons with either long axons as represented over here, which is coming from the brain to the spinal cord, right? Or short axons, which are residing in the pyramidal cells of the brain itself. So these are hand-drawn versions of these axons, which were drawn by Ramon Cajal. So he was a very famous neurophysiologist, right? There is a book now which has a compilation of all his hand drawings, The Beautiful Brain, right? The drawings of Santiago Ramon y Cajal. And the beauty of this scientist is that at the time that he was working, there was just this very primitive kind of microscope that used to be present, right? He used to take brain sections. He was one of the people who, along after Golgi had developed the silver staining technique, he was one of the people who used the silver staining in the brain to stain the neurons differentially so that it could be imaged under the microscope. And when he used to see that, he used to draw it simultaneously uh, on a paper, right? Because at that time, cameras were not like cameras attached to microscopes were not a thing, right? So he used to hand draw whatever he used to see, and based on just his observations of these stained brain sections, he made wonderful observations, which stand true even till date, right? We'll read more about it, or we'll study or discuss more about it in the second part of the session, wherein we are studying that alternate history, right? In which Santiago Ramon y Cajal has also been talked about. Okay. So, who first discovered important evidence about dopamine in the brain, right? Was it Arvid Carlson or Ole Hornikiewicz, Julius Axelrod, or Seymour Ketty, right? So, which one of these actually was more related to gathering evidence about dopamine? So, the clue is that that person also received a Nobel Prize for the same, right? And the person was Arvid Carlson. He not only discussed about dopamine and that it is present in the brain, but he also linked uh, it to the disease Parkinson's, right? And how the lack of dopamine in the brain could, or L-dopa in the brain could lead to Parkinson, right? So he also discussed about that portion. So then, question number 9 is, what are pheromones used for, right? Is it to make food or to help animals of the same species communicate or to build houses or to play games, right? So, the obvious answer over here is to help animals of the same species to communicate, right? The keywords over here being that they should be of the same species, right? That is the only way pheromones actually work. Okay. Now, this is an example of pheromones, right? So, for instance, we are not naming a compound over here, at least in this representation. For example, there are a set of pheromones, right? Uh, in the female, they might, uh, you know, uh, inhibit a certain pathway and excite a different kind of pathway, which might instigate the female to be sexually receptive towards a male. But if the same set of pheromone was in turn given to a male, right, it would probably inhibit the pathway that it was exciting in the case of female, and instead excite a pathway that was inhibited earlier, and this would lead to a different kind of a reaction in the male brain, which would instigate that if this pheromone is coming from a male, then this particular male whose brain this is should instigate a fight with them, right? Or they should mount, right? So it is a safe bet to say that this pheromone could be a male pheromone. That is why it showed this kind of a circuitry in case of female and this kind of a response in case of the male, right? Yeah. So, in case of humans, for example, it is still contradictory whether the concept of pheromones is applicable to humans or not. But there are like research and studies that talk about both the schools of thoughts that like pheromones do not play that much of an important role in case of humans, or they are important in certain aspects of human behavior or development, right? So both cases have been spoken about. So, but like scientifically, what we also know is that chemicals that appear to be human pheromones, again, the keyword is appear, right? So it has not been definitely stated that yes, this is a human pheromone, right? But the candidates could be androst androstenedione, right? And which is an odorous androgenic metabolite, and estratetraenol EST, which is an odorous estrogenic metabolite, right? So the androgenic metabolite is of course released by the male, and the estrogenic metabolite is of course released by the female, right? So this was an fMRI based, if I'm not wrong, response that was studied. Um and wherein different brain regions were mapped of these different sexuality of men and women to the response to androgen or EST, right? So, what was seen, for instance, was that heterosexual men did not respond at all to A and D, while heterosexual women responded to A and D, but their response was different from homosexual women, right? So, over here, you can see it is in this hippocampus thalamus region that this response is located in case of heterosexual women, but in case of homosexual women, the response to A and D is located in different part of the brain as opposed to this, right? But in homosexual men, actually, it is very close by to what was the response or what was the area that lit up in case of heterosexual women also, right? Similarly, when the response to EST was tested, it was seen that heterosexual men had a response to EST, which was this estra, tetra, and all, right? Again, heterosexual women also responded to it, but it was in the forebrain region, so it is probably away from this scope of, sexual attraction. And okay, the context to this is that female brain is more sexually dimorphic than male brain. You can study more about it in any neuroscience book, right? So, which is why it has a response in this case also. And again, you can see that to EST, there were multiple responses that were seen, but most of them differed in the regions at where the response had originated, right? So again, it is contradictory in case of humans, whether it works or not. Question number 10 is, where did scientists first measure electrical activity to study pheromones, right? In mouse brain, in cat tails, in insect antenna, or in human noses, right? So it was actually measured in insect antenna, which is one of the model systems in which it has been safely and successfully established that yes, the concept of pheromones is present, right? Okay, so before moving on to that history part, while I was going through Candle, the neural science book, right? What I also came across was this very visual and very good representation of Roger Sperry's experiment, which we had discussed in one of Professor Varadhan's lecture, right? So over here you will be able to visualize it better because it is represented in such a beautiful way that what he had actually done, if you remember, was that this is the case of a normal frog, right? In case of a normal frog, the eye has been color coded like this, right? And you can see these are the normal connections that it is making to the brain, right? And the action to this connection is that when it sees a fly, it can actually eat that fly, right? But in case of Roger Sperry, what he had done is, if you remember, right, he had first broken these connections, right? He had severed these connections and also he had rotated the eye by 180 degrees, right? So you can see over here that this yellow goes to the bottom, blue comes to the top, red goes to the back and green comes to the front, right? Over here. So after that, what he observed was that the reinnervation happened, right? So this P region was going to this A region. Still, this P region went to this A region. This A went to this P. Similar case still happened, right? And similarly, this D was connected to V, which it still was, and the V was connected to D, right? But over here, since I was also inverted to 180 degree, what happened is its field of vision changed, right? So earlier, over here, you can see the retina used to see it like this, right? Still, the vision is over here only. But the processing in the brain region, that is the tectum, that changed, right? So it was actually supposed to invert whatever the retina was seeing so that it will give us an upright image. So if you are aware of the concept, when we study physics also, we know that there is a virtual image, there is a real image, right? And the image that we get on our retina or when we see is an inverted image actually. And when the brain processing is done, then this inverted image is actually uh, inverted so that we see it as an upright or a standing image, right? So that is the normal way that this happens. But in this case, when I was inverted, what happened was that the retina still formed the correct image, right? But then at the level of processing in the brain, which is the tectum region of the brain, what happened was, sorry, that this image was not inverted, right? So the animal still, thought that it was either upside down or it could not visualize this particular subject as is in the field of view, right? So that is why instead of aiming towards the fly, it aimed away from the fly because the image formed in its brain was like that, right? So, if you want to read about what was written in this legend, it is said that in case of this normal system, the frog lens projects an inverted visual image into onto the retina, and the optic nerve then transfers the image with an additional inversion to the optic tectum. The orderly arrangement of inputs to the tectum is responsible for this transfer. Neurons in the anterior retina project axons to the posterior tectum, while neurons in the posterior retina project axons to the anterior rectum. This is a P and A, right? Similarly, the dorsal and ventral, how they are projected, that is written, right? As a result, visually guided behaviors are accurate, such as catching of this fly, right? But if the optic nerve is cut and the eye is surgically rotated in its socket before the nerve regenerates, visually guided behavior is aberrant. When Y is present overhead, the frog perceives it as below and vice versa. The inversion of behavioral reflexes over here, it should be, results from the connection of the regenerating retinal axons to their original targets, even though these connections now transfer an inverted, inappropriate map of the world to the brain. Right? So the targets are still correct. Just this processing has gone haywire, right? Because of the inversion. So this is it for the first part of this lecture. I'll just stop sharing and reshare for the second presentation. Oh, it has not gone as yet. Okay, I've been able to change it. Yeah. So far, was there any doubt in what we have gone through, or anything you would like to have additional knowledge about? If not, we'll just switch to the other presentation. Yeah. I'm not sure if it is not being shared.
I think we are clear so far. Thank you. We can move on.
Okay. Yeah. Yeah. Uh, so I had come on to the slide, but I had forgot to share. Right. So, as I told you, at least this week, what we'll be doing is we'll go through an alternative classical history of neuroscience, right? Just to clear some other basics of neuroscience that you might have missed, which you might require, for your future understanding of this course. So that is what we'll be doing. Most of what has been kept in the slide is from this book Principle of Neural Science by Candle. Some of it is from Student's Guide to Cognitive Neuroscience by Jamie Ward, and the other resources are mentioned on the slides as well. Right. So over here we are going only to the classical history, some part of the modern history, and we'll not be touching the contemporary history as of now, right? Once you are through with the subject, maybe then, if it is required, we can come back and we can discuss the contemporary history, which is a very recent history in or advances in the field of neuroscience, right? So we go way back, right? Not in the 1900s, not in the 1800s, but way back to the second century, right? Wherein there was this guy named Galen, right? He was a Greek physician who basically proposed that nerves convey fluid secreted by the brain and spinal cord to the body's periphery, right? So what he thought was that there were these nerves that were coming out of the brain, which were bringing some fluid from the brain, and they were taking the fluid to different regions of the body, right? And that is how the brain was actually monitoring or driving the entire body. That is what he thought. We are not saying what he thought is correct, right? A lot of this was thought of as correct or thought of as possibly the truth when it was told, but it was later either accepted or denied, right? Rejected. So this was one of the things that was not true. But what insight it gave to us was that Galen, even in the second century, believed that that brain controlled the body, right? How it controlled the body was not clear, or at least he did not get to the correct understanding of how this happened, right? So his views dominated Western medicine until the microscope revealed the true structure of the cells in the nervous tissue, right? And what he said was widely believed in the West. Okay. So in the early 1800s, now we are going, and 1800s in general, nervous tissue did not become the subject of special science until the late 1800s. Okay, so this is towards 1880s, 1890s when Camillo Golgi and Santiago Ramon y Cajal produced detailed, accurate descriptions of the nerve cells. Now, what they had done was first comes Golgi, right? So this is not the Golgi apparatus. This is the Camillo, okay, it is the Golgi apparatus guy also. But over here in neuroscience, why Golgi is relevant as a neuroscientist is because he developed a method of staining the neurons with silver salts that reveal their entire cell under the microscope, right? Cell structure under the microscope. Now, before, okay, I have accidentally switched off my video. I'll just reshare my screen. Yeah. So before understanding what he had done, how he had done it, first let us understand the need for this kind of a method to be developed, right? So before Golgi and before Ramoni Cajal, what happened was that there was an entire brain. be it a human brain, be it a mice brain, dog brain, cat brain. So back in 1800s, ethical science was not a thing, right? uh, Scientists, physiologists, doctors, they used to do research on whatever they used to get their hands on, right? So that is why a lot of ethical conundrums had uh, were present around that age, right? But what happened was, even if you have a brain, you can only study so much of the brain by looking at it, right? And if you even, how will you assess the finer structures within the brain, right? So, how could you do it? So, what the scientists started doing was that they thought, again, if they took reference from botanists and how the botanists used to look at flowers or how the botanists used to look at seeds was that they used to, chop it really finely, and then they used to put it under the microscope, and then they could visualize structures very nicely, right? So what these people started doing was they used to chop the brain finely into very thin slices, and they put it under the microscope and they began to look at it, right? Now, if any one of you have actually seen even sections of the brain, right? So I could have actually kept a slide over here. uh, so what it looks like under the microscope, even very thin sections, what they look like is you will be able to make out distinct regions of the brain, but you will not be able to make out any cellular details within the brain, right? So that in if you just section the brain and you put it on this under the microscope, that is what you can see, right? You can identify, okay, this is the hippocampus, this is the cortex, this is the cerebellum, etc., etc. But you'll not be able to tell what are the cells within those regions, right? So that was the challenge that these scientists were now facing. So now, then what happened was that they wanted to understand how they could specifically look at neurons, right? Now, this was the time when antibodies were also not a thing, right? Antibodies. Now, how will we look at something? If I want to look at a neuronal cell, for instance, I will just stain my section with new N antibody, which will stain the neuron, and I can then put a fluorophore that goes and binds to my antibody. That is a protein. Antibody is a protein, right? So which will recognize only my neurons in the tissue, right? And then this fluorophore will give me fluorescence, and it will just fluoresce at the places where neurons are present, right? Similarly, if I have to look at some other cell, glia, for instance, in the brain, astroglia, if I have to look at astrocytes, right? So again, I will take an antibody against a protein that is specific to astrocytes. I will label it using that protein, which is again the antibody staining that now we are very well aware of and we use so extensively, right? So that was also not available back in the 1800s, right? So what they did was they wanted to come up with a method through which they could stain the tissue in such a way that neurons could be visualized. So while trying to develop multiple such dyes and stains, Golgi finally developed a method of staining these neurons with silver salts, right? So, silver staining, which is the silver nitrate staining, is also widely, even currently, done in labs, right? So, it gives us a morphology of the neurons in the brain, for instance, right? So, he developed this method through which now he could look at the neurons, particularly of the brain under the microscope, right? Now he could see clearly that each neuron typically has a cell body and two types of processes, branching dendrites at one end and long cable-like axon at the other, right? So this he could correctly make out that there was a cell body, right? Near the cell body, there were multiple branches that were present, which were the dendrites that had branched out, and there were long, single long processes that came out of the cell body, which were known as axons, right? So this he could make out, right? So these are from original texts of Golgi. Based on what he stated and what he observed, what theory Camillo Golgi came up with was the reticular theory, right? What he said in reticular theory was that nervous system is composed of a single continuous network of cells rather than separate nerve cells, right? So, for instance, in B, if you see, he said that this is a single continuous network of cells. It is not different nerve cells altogether, right? However, this is incorrect, right? It was later proven that this theory was incorrect. The theory was proposed by Joseph von Gerlach in 1871 and it was popularized by Camillo Golgi, right? Then came Ramon y Cajal, Santiago Ramon y Cajal, right? So, what he was a Spanish researcher. What he did was he used Golgi's technique of silver staining, and he discovered the nervous system is not a syncytium, that is a continuous web of elements, but instead it is a network of discrete cells. So he said that no, it is not, the reticular theory is not true, right? It is not a network or a single network or a single mesh, right? There are discrete nerve cells that are present that form this nervous system. In the course of this work, Ramon Cajal developed some key concepts and much of the early evidence for neuron doctrine, which basically is the principle that individual neurons are elementary building blocks and signaling elements of the nervous system. Right? So the neuron doctrine was actually given by, well, its initial uh, uh, proposition was based on the work of Ramon Cajal, right? So again, this is the book I just showed to you, The Beautiful Brain, right? And these are actually hand drawings of Ramoni Cajal, right? So as I explained to you at that time, cameras attached to microscope was not a thing. So whatever he used to see, he used to draw, right? Over here you can see how he has even drawn different kinds of neurons, right? It was due to his studies that we came to know that okay, there are these kinds of neurons that look like a tree. There are these kinds of neurons which look like have a pyramidal kind of cell body. That is why they were known as pyramidal neurons, right? So these understandings or these terminology also came because of his studies, right? Now, what is interesting is that both Cajal and Golgi were actually awarded the Nobel Prize, although they had competing theories, right? The theory that was proposed by Cajal was actually correct. Golgi's theory was incorrect, but Golgi had developed the technique due to which these findings could be made and neuroscience could be advanced. That is why both of them together were awarded the Nobel Prize uh, for physiology at that time, right? Uh, then comes Ross Harrison, right? So you remember Ross Harrison from Professor Varadhan's videos also. He was an American embryologist who provided support for the neuron doctrine in the 1920s, right? What he showed was that dendrites and axon grow from the cell body and they do so even when each neuron is isolated from others in the tissue culture, right? So even when they were isolating separate cells, still dendrites and axons used to grow from the cell body, right? So this basically affirms that they are separate entities, separate units, right? Harrison also confirmed Ramoni Cajal's suggestion that the tip of the axon gives rise to an expansion, which is the growth cone, which is the axonal growth cone basically, which leads to the developing axon to its target. either to other nerve cells or to muscles, right? So he also confirmed his axonal growth cone hypothesis. Thereafter came another scientist in the mid 1950s. He was Sanford Palek, right? He provided the final and definite proof of the neuron doctrine in mid 1950s after the introduction of electron microscopy, right? So at this time, electron microscopy had also been introduced. What he found was that in this landmark study by him, he unambiguously demonstrated, that is, without a doubt, he demonstrated the existence of synapses, specialized regions that permit chemical or electrical signaling between the neurons, right? Then, again, we are going back in history again for another thought process to evolve, which was Luigi Galvani, right? Who was a physician as well as a physicist, right? So currently, like if he would have been a physician and physicist right now, he would have been an interdisciplinary scientist. So even back in the 1700s, that was the case, right? So what he discovered was that muscle and nerve cells produce electricity, right? So that was a discovery made by Luigi Galvani in the late 1700s, right? So they had some electric potential, is what he found. This led to the beginning of physiological investigation of the nervous system, right? Okay. So later, one of his experiments went really, uh, they have been studied through thoroughly, sorry. It is a Galvani frog experiment, right? Just give me a moment. I have a little congestion. I'll just reshare my screen. Yeah. So this was the experiment in which a frog that had already been dead for a while was basically pinned to a particular board, dissection board. And what happened was accidentally, while they were moving some wires, one of the wires, it hit or it touched the hind limb of the frog that had already been opened up and dissected and worked on, right? As soon as this wire touched the limb of the frog, what happened was that the limb twitched, right? So, like, even though the frog was dead, it twitched as if, it was a normal muscular event or normal muscular motion. And as you know, that the hind limbs of the frog are very strong, right? Because they can jump, it provides that strength. So they are really rigid, tough, and a lot of um that momentum for jump it has, right? So it was behaving once this wire was being applied to the, or this current was being applied to the hind limb of the frog, the natural kind of muscle movement was being observed even in this dead dissected frog, right? So that is how he basically understood that there is some kind of an electric potential within the nerve cells and the muscle cells that this actually happens within the system of the frog also, right? This kind of an event happens. Thereafter came the studies by Johannes Muller, Emil du Bois-Reymond, and Hermann von Helmholtz, right? They were German physiologists who succeeded in measuring the speed of conduction of electrical activity along the axon of the nerve cell, and they further showed that the electrical activity of one nerve cell affects the activity of an adjacent cell in predictable ways, right? And their work later inspired the modern electrophysiology in the 19th century, that is the 1800s. So this was also pre 1800s or in the early 1800s, right? Thereafter came Johannes Peter Muller, who gave the doctrine of specific nerve energies. Now it is known as the law of specific nerve energies in 1835, right? A doctrine, once it is proven, it becomes a law. Sorry. It basically states that nature of perception is defined by the pathway over which the sensory information is carried, right? The mind has access not to objects in the world, but only to our nerves, right? Later, there were experiments done by Penfield, which once you will study, you will have a better understanding or you will be able to visualize what Peter Muller said back in 1835. Uh, Prior to that, 1835, it had already become a law, right? Emile du Bois-Reymond, who is renowned for co-discovering nerve action potentials and pioneering experimental electrophysiology, uh, was Muller's assistant in Department of Anatomy and Physiology at the University of Berlin in Germany. And Hermann von Helmholtz, that we talked about, in 1849, he measured the speed that electricity flows in a frog sciatic nerve. That is the nerve that is uh, connecting the basically thigh muscle to the spinal cord. That is the sciatic nerve, right? By stimulating it electrically at one end and measuring the electrical signal at the other end. So by this time, you can safely understand or have an understanding of the concept that it was understood that there was this electrical potential that did exist in case of the neurons, right? So pharmacology made its first impact on our understanding of nervous system and behavior at the end of the 19th century when Claude Bernard, uh, Paul Ehrlich, and John Langley in England demonstrated that drugs do not act just anywhere on a cell but rather bind discrete receptors typically located in the surface of membrane of the cell. This insight led to discovery that nerve cells can communicate with each other by chemical means also, right? So these people we have studied in detail also in Sir's course as well, how each of them contributed to our understanding, pharmacologically speaking, right? So, psychological thinking about behavior dates back to the beginning of Western science when the ancient Greek philosophers speculated about the causes of behavior and relation of the mind to the brain. Mhm. Okay, accidentally, this is what I keep doing. Yeah. Then comes Rene Descartes, right? Again, another line of history again we are taking. So we talked about one line of history wherein how neurons are present, whether neuron is a single, functional entity of a unit of the brain or not. That theory was being followed, right? There was a separate one in which electrophysiology was being understood. There was a third one in which the electric potential in the neuron was being understood. And then there is this other parallel history in which there is this body and mind debate that is going on, right? So Rene Descartes, he was actually a mathematician, right? So there is some Descartes theorem in maths also that is studied. So he was actually a mathematician only, and he was a good mathematician, but not so great in neurology, neurophysiology whatsoever. So in 17th century, what he said was he distinguished body and mind. That body and mind are two separate entities, right? What he said was that there is he had a dualist view that these are two separate entities, and he said that brain mediates perception, motor acts, memory, appetites, and passion, everything that can be found in the lower animals, but mind, the higher mental functions, conscious experience, characteristic of human behavior, is not represented in brain or any other part of body but in the soul, a spiritual entity that communicates with the machinery of the brain by means of the pineal gland, this gland, which is a tiny structure in the midline of the brain. Right? Now, why did he say this? Right? He said this only and solely based on the fact that he saw that brain, is uh, what is that term known as? Oh, there is such a simple term I'm missing. Bilaterally symmetrical. Okay. So that the brain, like a lot of other organs in our body or features in our body, brain is bilaterally symmetrical. That is, that one part of the brain resembles the other part of the brain. Right. So there is a forebrain hippocampus, a part of the cerebrum, part of the brain stem, hypothalamus, pituitary gland, everything is present in part of the one side of the brain also, and then on the other side of the brain also, right? And they are identical to one another, structurally identical. I'm not saying they have the exact same function or something. They are structurally identical to one another. That is, if we slice the brain into half in this way along the corpus callosum, we will have two identical halves of the brain, right? What he thought was that all these other body parts, all these other parts of the brain, hypothalamus, cerebrum, cerebellum, thalamus, pituitary, brainstem, etc., all of them were present in duplicate structures, right? Like one half was over here, one half was over here. But pineal gland was a single entity which was present in the middle, right? Is what he thought, which is why he said that this was the region through which that higher entity, that was the soul, was communicating with the brain. Right. However, that was not true. Later, it was found out that pineal gland also it is present towards the center, but again, it has two halves. Right. One goes into this part of the brain, other goes into that part of the brain. So that was not true at all. So, as I said, good mathematician, not a great neuroscientist, right? So this is our current understanding of the pineal gland that yes, indeed, there is a two pineal gland, two parts of pineal gland also. But at that time, he couldn't visualize that. And even if he did visualize that, the other parts also that he said were not true, right? Then comes Baruch Spinoza, right? Later in 17th century, he began to develop a unified view of the mind and body. Uh, then came like unified view of mind and body means that it is the same entity, right? So consciousness and motor skill, perception, etc. They reside in the same unit, which is our current understanding also. So this basically fueled a debate between empiricists versus idealists, right? So empiricists believe that brain is initially a blank state, right? So when we are born, there is nothing that we know of. It is later filled by sensory experience, whereas idealists, notably Immanuel Kant, right? Again, Immanuel Kant, if you remember, he was a good immunologist, right? He believes that our perception of the world is determined by inherent features of our mind or brain, right? It is still a debate. It should not ideally be a blank state per se. Uh, there are some, but we don't know, right? It is an open debate even today. Then, in the mid 19th century, Charles Darwin set the stage for the modern understanding of the brain as the seat of all behavior, right? So he also advanced the even more radical idea that animals could serve as models of human behavior. Again, as I said, these were the times when ethics related to research per se were not a thing, right? So yes, humans could be models, not just of behavior, but of experimental studies as well. Thus, the study of evolution gave rise to ethology, the investigation of behavior of animals in their natural setting, and later to experimental psychology and study of human and animal behavior under controlled conditions, right? So this is Charles Darwin. Later, at the beginning of 20th century, that is in 1900s, Sigmund Freud introduced psychoanalysis. As the first systematic cognitive psychology, psychoanalysis framed the enormous problem that confront us in understanding the human mind. Right? So how like psychoanalysis currently it's used as in a little bit of a negative way that why are you psychoanalyzing me like that kind of a way, right? So there were attempts to join biological and psychological concepts in the study of behavior, which began as early as 1800 when Franz Joseph Gall, a Viennese physician and neuroanatomist, proposed two radically new ideas. First, he advocated the brain is the organ of the mind, that is, all mental functions emanate from the brain, right? So first part was actually true. In doing so, he rejected the idea that the mind and body are separate entities. Second, he argued that the cerebral cortex did not function as a single organ but contained within it many organs that particular regions of cerebral cortex control specific functions. He was partly correct, right? Cerebral cortex has multiple different features, multiple different subdivisions such as thalamus, hypothalamus, hippocampus, amygdala, basal ganglia, etc., right? But it is not entirely true that particular regions are responsible for specific functions, right? Because a lot of functions, and especially higher order functions and cognitive functions, are an amalgamation of a lot of regions working together, right? Yeah. So over till here also he was a little bit correct. But later what he said was he enumerated at least 27 distinct regions or organs of the cerebral cortex. Later, many more of these were added, each corresponding to a specific mental faculty. This is where he went really, really wrong, right? So, what he said was he assigned intellectual processes such as the ability to evaluate casualty, to calculate causality, to calculate and to sense order to the front of the brain, right? So, what he basically said was that he developed a new kind of science, or it was actually a pseudoscience, wherein what he said was that assessing the skull of a person, you can understand how the person is, right? So, for instance, the people who have a smooth and smooth region over here, they are very cautious, but if the skull is a little dented over here, then they are not very cautious, right? If they have a for instance bigger forehead, then they are of a certain nature. If they have a smaller forehead, they are of a certain different kind of a nature. This is the kind of thing he got into, which is not true, right? From the surface of the skull, you cannot link these features to a person, right? So, instinctive characteristics such as romantic love, amativeness, combativeness were assigned to back of the brain over here, right? Yeah. Even most abstract of human behavior, such as generosity, religiosity, secretiveness, were assigned a spot in the brain. Now, that is not normal, right? Although Gall's theory of localization was prescient, His experimental approach to localization was extremely naive, right? Influenced by physiognomy, the popular science based on the idea that facial features reveal character. Gall believed that bumps and ridges on the skull of people well endowed with specific faculties identified the centers for those faculties in the brain. So how he came up with this was that, for instance, there is a person who is very rude, right? And that person has a dent in certain region of the skull, right? The skull is not smooth, right? There are bumps and dents across the skull. Like it is just normal. It is not like there's this a hole there, but it is not smooth, is what I'm saying, right? So if that person had a dent in a certain region, he applied that, region to, being polite. That since this person has a dent in this region, this person is rude. So this is how he came up with this kind of a map, right? So he assumed that size of an area of the brain was related to the mental faculty represented in that area. And Gall's studies of localized brain functions led to phrenology, a discipline concerned with determining personality and character based on detailed shape of the skull. It was a pseudoscience. We can forget about it now. In the late 1820s, Gall's ideas were subjected to experimental analysis by French physiologist Pierre Florence. Now, how would he have done experimental analysis? Imagine, right? What he did was he systematically destroyed Gall's functional centers in the brains of experimental animals, right? He attempted to isolate the contribution of each cerebral organ to behavior. From these experiments, Florence concluded that specific regions of the brain are not responsible for specific behaviors, but all brain regions, especially the cerebral hemispheres of forebrain, participate in every mental operation. Now, that is also an exaggeration, right? Any part of cerebral hemisphere Florence proposed is able to perform all hemispheres' functions. That is not true. Injury to any one area of the cerebral hemisphere should therefore affect all higher functions equally. Again, that is not true. This was popularized as the holistic view of the mind. So, opposing views were coming up in that period, right? Then the holistic view was seriously challenged, however, in the mid 19th century by the French neurologist Paul Pierre Broca. If you remember in the, okay, you guys had not attended the first uh, lecture that I had conducted, and I have still not uploaded its recording. I will shortly. So in that I have actually described this Broca's region, Wernicke's region, how one is responsible for speech articulation, the other is responsible for speech production, right? And uh, how that gave us a lot of insight about the brain, right? And British neurologist Hugh Links Jackson, right? So these were the people disregarded this holistic view. So regional studies of Broca, Wernicke, and Jackson were extended to cellular levels by Charles Sherrington and Ramoni Kahal. So Charles Sherrington, again, you would recall him from Sir's lectures, who championed the view of brain function called cellular connectionism, right? So this is Charles Sherrington. According to this view, individual neurons are signaling units of the brain. They are arranged in functional groups and connected to one another in a precise fashion. Wernicke's work and that of French neurologist Jules de in particular revealed the different behaviors are produced by different interconnected brain regions, right? Thereafter, uh, okay, I have it. It does have a little bit of understanding about that Broca's region, Wernicke's region, I think, right? Although phrenology was fatally flawed, the basic idea of different parts of the brain serving different functions paved way for future development in the 19th century. The most notable of which are Broca's reports of two brain damaged patients, right? So these were patients who had gone through some accident due to which certain regions of their brain were damaged, right? So he had reported it as a case study. in 1861. He documented two cases in which acquired brain damage had impaired the ability to speak but left other aspects of cognition relatively intact. Right? He concluded that language could be localized to particular region of the brain. Okay. So first case was in 1861, he described a patient, Libogne, right, who, as a result of stroke, could not speak, although he could understand the language perfectly well. Yeah. This patient had no motor deficits of the tongue, mouth, or vocal cords that would affect his ability to speak. So he could understand language. His motor functions were intact of the tongue, mouth, vocal cords. How is that assessed? Because tongue, mouth are also required for eating, chewing, those kinds of other kinds of functions, right? Blurting out something or just calling out, right? So vocal cords also can be assessed whether it is working or not, because otherwise the sound will only not come, right? But that was also fine. Motor deficits were not there in these regions as well. But he could utter isolated words, he could utter isolated words, whistle, and sing a melody without difficulty. But he could not speak grammatically or create complete sentences, nor could he express ideas in writing. So what used to happen was that this patient had a part of his brain damaged, right? Now this part, it was later identified, is responsible for speech production, right? Just a moment. Speech production is what it is doing, right? So the person can understand what you are saying to them. The person can assess and can, form, like understand what is being said. He can understand that what does it mean, what does the person have to do, all that he can make out in his head, right? But he cannot communicate because the words don't come out of the mouth. In that order, right? The speech production is what was affected over here. So later, postmortem examination of patient's brain showed a lesion in the posterior region of the frontal lobe, which is now called as Broca's area. Right? So more about this I have already discussed in the previous session. Then came Carl Wernick, right? He took next step in 1876 and published a now classic paper, The Symptom Complex of Aphasia, a psychological study on an anatomical basis. What he described in this was another type of aphasia, failure of comprehension rather than speech, a receptive as opposed to expressive malfunction. Now, in his case, what happened? He had another patient who again had a different region of the brain wherein there was an injury, right? Due to which what used to happen was the person can speak, but once the person speaks, he blurts out like anything. He like and when you speak to the person will not be able to make out what exactly you are trying to communicate, right? So the region of the brain that this person had affected had later been associated with speech articulation, that whatever is being said can be heard, it can be taken into the brain, but it cannot be processed simultaneously. Because it cannot be processed, the person can also not process his own thoughts. So he, like, he speaks anything. Speech production is there, but speech articulation is not there. Right? Whereas Broca's patient could understand language but not speak, Wernick's patient could form words but could not understand language. Right? Moreover, locus of this new type of aphasia was different from that described by Broca. The lesion occurred in the posterior part of the cortex, where temporal lobe meets the parietal and occipital lobes. Right? So these lobes you will eventually understand. So this is the Broca's area, this is the Wernick's area. Right? Subsequent studies argued the language itself was not a single entity but could be further subdivided into speech recognition, speech production, and conceptual knowledge. Right? So it eventually was a result of the studies of Broca and Wernick that these kinds of subclassifications could actually be made. This was motivated by the observation that brain damage can lead either to poor speech comprehension or and good production, or good speech comprehension and poor production, right? Then came Corbinian Brodmann. Again, Brodmann was an anatomist in Germany. He led a new school of thought that cortical localization at the beginning of 20th century, one that distinguished functional areas of cortex based on shape of the cells and variation in their layered arrangement, right? So this is what he had drawn, right? Using the cytoarchitectonic method, Brodmann distinguished 52 anatomically and functionally distinct areas in the human cerebral cortex. However, this was also not correct, right? Lashley later was deeply skeptical of the cytoarchitectonic approach to the functional mapping of the cortex. He, and after him, many other psychologists concluded the learning that other higher mental functions have no special locus in the brain and consequently cannot be attributed to specific collections of neurons. He reformulated the aggregate field of view by further minimizing the role of individual neurons, specific neuronal connections, and even specific brain regions in the production of specific behavior. So, this was going the other way now. According to Lashley's theories of mass action, it is the full mass of the brain, not its regional components, that is crucial to function, right? Then comes Wilder Penfield. So, although it is questionable what he had actually done, sorry, but the kind of understanding of different regions of the brain that we have today is mainly because of studies of Wilder Penfield, right? So, we'll study this person. Just give me a very short break. Yeah, I'll share my screen again. Uh, yeah. Okay. So, between 1928 and 1947, Penfield and colleagues carried out a series of remarkable experiments on over 400 living human brains. This is why it is a little questionable, right? So, this was documented by Penfield and Rasmussen in 1950, right? Now, the patients in questions were undergoing brain surgery for epilepsy, right? So he was a neurosurgeon actually. Penfield was a Canadian neurosurgeon, and he used to treat a lot of epilepsy patients, right? And back then, epilepsy usually was treated by lobotomy, which is basically removal of a region of the brain which is causing seizures, right? So before that removal, the brain region had to be identified so that only that minimal region is removed and there is not much damage to the patient, right? And if it is a very crucial region of the brain which is causing epilepsy, such as a region that is responsible for, say, respiration, right? Then the cost to benefit ratio would not allow removal or lobotomy of that region. But if, say, that region is responsible for speech production, then because seizure is also a very, difficult condition to live with, maybe it could be considered that, okay, lobotomy of this region can be done. Up to some extent, the person will be able to speak again after some training of the nervous system again, right? So that was the scenario back when he was experimenting, right? To identify and spare regions of the brain involved in movement and sensation, Penfield electrically stimulated regions of the cortex while the patient was still conscious, right? So if you know, okay, it is written over here also, procedure was not painful, but the patient did report some fascinating experiences. So, what I was saying is, if you know, the brain itself does not have any pain receptors, right? So, for instance, generally, even today, when a neurosurgery is being done, the patient is kept awake, right? The patient is anesthetized for a little while during the time the skull is getting drilled to form that window through which the brain can be accessed, because the skull will have nerve endings that will cause pain to the person. But once the brain is actually reached, the brain doesn't have any pain sensation, right? So even if you uh, poke around with a knife in the brain, your brain will not feel, you will not feel any pain, right? And usually the patients are made to stay awake during these kinds of neurosurgeries, so that it can be assessed that you know you are targeting the correct region for the study, because if the incorrect region is targeted, the patient will feel something and they can just communicate that with you at that time itself, right? Rather than finding it out after the patient has come out of his anesthesia or his state of comatose, right? So it is better that simultaneously, if you can avoid those things, yeah, that is why this kind of a thing is done, right? So what were these fascinating experiences, right? So what he used to do, okay, before going into this, what he used to do, Penfield used to do was that now the brain of the patient is open, right? uh, he used to take an electrode and he used to poke it at different regions in the brain, right? Now, he was trying to identify the focal point of epilepsy only, but the interesting thing he did was that he used to record whatever was happening, right? For instance, if he poked this particular region of the brain and the patient reported that they felt something in their toes, right? They felt as if something that their toes were burning, right? He will write this down, right? If he suppose poked some other region of the brain and the patient started laughing, it would generally be due to that kind of a response getting stimulated in that patient, right? So these different kinds of experiences is what he noted down, and later he mapped the brain regions to different kinds of out to different kinds of reactions to different kinds of modalities, right? So, when stimulating the occipital lobe, one patient reported a star came down towards my nose, right? Upon stimulating a region near the central sulcus, another patient commented, those fingers and my thumb gave a jump, right? After temporal lobe stimulation, another patient claimed, I heard the music again. It is like the radio, right? So, it was probably involved with audio uh, sensory apparatus, right? She was later able to recall the tune she had heard and was absolutely convinced that there must have been a radio in the operating theater. So this is Penfield, and this is his drawing of the brain, right? Of course, the patients had no idea when the electrical stimulation was being applied, and they couldn't physically feel it or see it also, right? That is why it is ethically questionable. As far as they were concerned, an electrical stimulation applied to the brain felt pretty much like a mental or a cognitive event itself, right? So this is a small timeline for development of methods and findings relevant to cognitive neuroscience from phrenology to the present day, right? The first nerve fiber was described by Purkinje in around early 1800s. Then Broca, Vertex region, that language localization concept came. This is something we have not done. Then EEG as a tool was developed in early 1900s. After that, actually, action potential was discovered by that Hodgkin-Huxley experiment, and multiple other techniques were later developed: CT, MRI, PET, fMRI, Bolt, etc. Right? Post this, I had kept a small unit about parts of the neuron, the cell body dendrites, axons. I am going to skip this for now. I'm feeling very congested. Uh, Let me see if there was anything that I had also kept a little bit about neurotransmitters, right? At least these are the kinds of neurotransmitters and what they are uh, associated to currently, right? So I will discuss this maybe in the next lecture. That is the coming Monday, the next Monday, right? For now, I'll just stop sharing. Yeah. Okay. So I hope you guys did have a different understanding of neuroscience and how different kinds of the knowledge that we have about the nervous system to date actually developed over a period of time, right? We did a parallel history or class of classical neuroscience, right? Uh, if at all you have any questions regarding this, you can always ask it to me in the following class, that is next Monday, or you can reach out to me. I'm sorry, or you can reach out to me via mail also, right? I will try to respond. Uh, that is it for this particular lecture from my side. And I'll be ending this. I hope you understood it. Thank you for attending it for this long. And good night to all of you. I'll just end.